So in this lecture, we're going to shift from focusing on populations to focusing on communities and ecosystems. And this first lecture in this section is about community structure and function. So it covers the material in chapter 16 of your textbooks. So just briefly, I want to cover the lecture themes. So we're going to start by defining what is a community, then talk in a bit more detail about what community ecology means. So what do community ecologists study and why is it important? And then we're going to talk about ways that we can quantify different elements of community structure. So the relative abundance of different species, species diversity, other kinds of diversity as well, the effects of environmental complexity on communities, and the effects of disturbance on communities. So first of all, what is a community? A community is an association of interacting species inhabiting some defined area. And some communities can seem very simple, so deserts that might be dominated by a single desert shrub, or seemingly so, ranging all the way to incredibly complex communities like tropical rainforests. So some of the questions that community ecologists concern themselves with are what are the factors that drive these differences in diversity in these different areas? So community ecology concerns how the environment influences community structure, and we'll talk about different elements of community structure throughout this lecture. So community structure includes attributes like the number of species in an area, the relative abundance of those species, and species diversity. And these things can all be measured in different ways. Incidentally, community ecologists tend, as a rule, to restrict their studies to groups of species that all make their living in a similar way. And in animals, we call these guilds. So, for example, a community ecologist might study pollinators, and they might be many varied species, but we could think of those as the guild of pollinators. Similarly, ecologists might study grazing animals, and again, we could refer to this as a guild. In plants, we refer to these things as growth forms or functional groups. So an ecologist might study annual plants, or they might study long-lived trees. These are growth forms or functional groups. So one element of community structure that ecologists are particularly interested in is the relative abundance of species. So for example, you can see the top picture on the right there is a North American desert, and the landscape there is dominated by pretty much by one species, the creosote bush. So clearly there's a huge relative abundance of that species compared to other species in the community. And conversely, the bottom picture is a tropical rainforest. You can see this huge array, this diversity of species, with no one species dominating that community. And so dominance is another term that we use to describe this, and something that ecologists are interested in. Are there one or a few species that are substantially more abundant than other species in the community? And when we're measuring things like this, we can measure them in very different ways depending on the organisms that we're studying. So, for example, if we were studying large trees, we tend to use biomass as a measure. If we're studying mussels or barnacles in the intertidal zone, we might use percentage coverage, because that would be a much easier way of quantifying dominance in that situation. In other cases, if we're studying grazing ungulates, we would probably look at the number of individuals. So it all depends on what works best for your organism of study. What's been found over the years of studying relative abundance of species in communities is that there tend to be regularities in the relative abundance of species in communities that remain true regardless of what kind of organisms we're studying. And we'll talk about this a little bit in a bit more detail over the next few slides. So Frank Preston was an ecologist, a Canadian ecologist working in prairie systems in the 1950s and 60s. And he graphed the relative frequency of different species in prairie communities over that time. And he observed consistent patterns. And he came up with what he described as his distribution of commonness and rarity. So when he graphed the number of species on the y-axis with a log their relative abundance, he found these kind of normal or bell-shaped curves. So what they essentially show is that a very low number of species are very rare, a very low number of species are incredibly common, but most species have this intermediate abundance, and that's what generates this bell-shaped curve. What's particularly interesting about this is that this distribution, this normal distribution, holds true seemingly regardless of what kind of organism you're studying. So even when plants were studied, or when moth species, or when bird species were studied, their distribution of abundance appeared very similar. So as Frank Preston continued to study these relationships, one thing he found was that in some of his samples, when he graphed them, it would only produce part of that bell-shaped curve. So you can see the two graphs on the right, the top graph, there's slightly less of that curve being displayed. So that top graph has come from a collection of 87,000 moths in an area of Saskatoon, Saskatchewan. And the bottom graph comes from a sample of 300,000 moths from Lethbridge, Alberta. And so the larger sample of moths that he'd taken produced more of that graph. So what he's arguing is that to produce the full bell-shaped curve, you just need to sample very heavily in the area. So the more samples collected, the more of that curve you're going to see. And that rings pretty immediately true when you think about the more you sample individuals within a community, the more likely you are to find those very rare species. You're not going to get those very rare species in a very small sample. So to uncover the whole community and all of the information about that community, you do have to sample very heavily. So this commonly observed distribution of abundances of species within communities is known as the log normal distribution, because it has this bell-shaped or normal-shaped curve. And there have been lots of different theories as to what drives this distribution in so many communities. Is it random environmental variables, as May suggested in 1975? So this is the idea that this distribution comes about just as a kind of statistical artifact. Or Sugihara in 1980 suggested that it was all to do with species subdividing niche space. So the availability of different niches is what drives this distribution, and what means that there are few, what leads there to be very few entirely dominant species. Either way though, this tool allows us to predict the distribution of abundance among species and communities, so it remains a really important tool for community ecologists. So talking about the relative abundance of species within a community is a good way to lead into a discussion about how we measure diversity. So the most common way that we measure diversity in a community is by looking at species richness. And this is done so commonly that we just often refer to it as the letter S. So species richness is the number of species found in a community. But also relevant to diversity is species evenness. So that's the relative abundance of different species within a community. So if you look at the two forest communities below, both of those communities contain 5 different species. So their species richness is the same in both cases. But in the community on the right, species evenness is much higher than the community on the left. And I dare say if you were walking through either one of those forests, the one on the right would seem like a much more diverse place. And so ecologists have spent a lot of time trying to work out ways to integrate these two measures of diversity, species richness and species evenness. So the most famous and commonly used model that integrates species richness with species evenness is the Shannon-Weiner index, and that's calculated by this equation below that calculates H, this integrated measure of diversity. And that's where pi is the proportion of each species in the community, and log E is the natural logarithm of pi. So this table shows the process by which you can calculate the Shannon-Weiner index for those two different forest communities. So there are two tables on the bottom figure. They list the different species in the communities. Then the second column is the number of individuals of each species, then the proportion of each species within the community, then the natural logarithm of that proportion, and then the final column is the multiplication of the proportion of the species in the community times the log of that proportion. And the sum of that final column is the Shannon-Weiner index. So what you can see is that the community on the left that has the same number of species but lower evenness has a Shannon-Weiner index value of 0.662, and the community on the right, which has greater evenness, has a Shannon-Weiner index of 1.610. So the Shannon-Weiner index acknowledges that community on the right-hand side is more diverse than the community on the left, by virtue of the fact that it has greater species evenness. It should be said that the lowest value that you can have on the Shannon-Weiner index is 0. That would be the value if you had a monoculture containing just one species. So we'll spend some time in class actually calculating the Shannon-Weiner index for these sample communities. So you can see here the community on the left is an insect community made up of 8 individuals and 3 species, but that community is dominated largely by this one large butterfly species. The community on the right is a community of insects made up of 8 individuals and only 2 species, but it has greater evenness with both of those 2 species represented by the same number of individuals. So it's interesting to think how the Shannon-Weiner index will interpret those 2 communities. So if we calculate the Shannon-Weiner values for both of those 2 communities, you can see the first one with 3 insect species comes out at 0.733. And the second community with only 2 insect species comes out at 0.693. So not too dissimilar in their Shannon-Weiner values. And that's because the second community, despite having 1 less species, almost compensates for that by its increase in species evenness. So you can see how effectively this model integrates those 2 different measures of diversity. So the Shannon-Weiner index is a really useful way of reducing this really complex idea of diversity down to 1 value. But depending on what we're studying, we may still sometimes be interested in these different measures of diversity separately. So species richness is still a really important value, and it's simply a question of counting the different number of species within a community. And while very simplistic, it's often the most important statistic in any biodiversity study. So species evenness can also be measured independently, and the most common way of doing that is by using a model called Pylus-J, which is a model devised by a Canadian ecologist, Evelyn Pylus. So in this equation, H obviously means the Shannon-Weiner index value, and H max is the Shannon-Weiner value, assuming that all the species in the community are exactly evenly distributed. So it's the total possible H value for the number of species in the sample. And that's the same as the log of S, so the log of species richness. So as the species evenness in the community you're studying increases, the value of J gets closer and closer to 0. Mathematical models like the Shannon-Weiner index and Pylus-J are really good ways for us to estimate and understand elements of community structure. But another tool we use to do this, particularly to look at the distribution of species abundance within a community, is to construct rank abundance curves. So this is a plot of the relative abundance of species against their rank in abundance. And so if you look at the graph below, you can see on the x-axis, you have a list of the rank of the species in the community. And then on the y-axis, you have relative abundance of those species. So the most abundant species in the community is on the left-hand side, and it slowly works its way down to the least abundant species on the right. So this is a really useful tool because it allows us to understand something about the distribution of abundances within the community just at a single glance. So this line tells us an awful lot about how that community is structured. So to take a simple example, we can construct rank abundance curves for those two hypothetical forest communities that we looked at previously. So the less diverse or the less even community, which is the picture on the top, is depicted by line B. It's a straight line because all 5 species in that community have the same proportional abundance. Whereas the more diverse or even community, which is the picture on the bottom, is depicted by community A or line A. So you can see this huge disparity between the abundance of species 1 compared to the other 4 species in the community. So this is a neat way to show that in rank abundance curves, communities with greater evenness are indicated by a lower slope or a flat line in this case, and communities with lower evenness or greater dominance of a few species have steeper slopes. So to give you a good example of when rank abundance curves have been used to good effect, this is a study by Eric Lamb, who studied or collected species abundance data in boreal forests, both forests that had burned recently within the last 4 years, and forests that showed no signs of having recently burned. And his data are represented by the 2 lines on the right hand side. The blue line represents the unburned sites, and the red line is a rank abundance curve of the burned sites. And what they show is that the most abundant species in those forests occur roughly evenly in the burned and unburned sites. But as you move down the ranks to the rarer species, in the burned sites, those rare species are represented less and less. So although these fires occur naturally, and occasionally they're set deliberately by humans, and that's to prevent fires burning out of control when they inevitably do happen. But what this study shows is that if we want those rarer species to be represented, we need to leave bigger gaps between fires, certainly more than 4 years. So far in this lecture, we've talked a lot about how we measure abundance and diversity in communities. What I want to focus on for a few slides now is how the environment affects communities. And specifically I want to think about the effects of environmental complexity on communities. So in general, species diversity increases with environmental complexity or heterogeneity. So heterogeneity is the quality or state of being diverse in character or content. So what this means is that as a landscape, for example, becomes more diverse and patchy, it's likely to support more species than a completely uniform landscape. But this idea of heterogeneity promoting diversity has been a complicated one for ecologists. So if you look at an environment like this tropical rainforest, you see this huge diversity of plant life. So it's really easy to imagine how insects, for example, could have become hugely diverse off the back of this. So there's so many opportunities for insects to specialize on different plants or to take advantage of different niche space that becomes available in a landscape that's as diverse and varied as this one. But a big question is, how did that diversity or that level of diversity of plants occur in the first place? What's the heterogeneity in the landscape that allowed that to happen? And it's that question that Evelyn Hutchinson in 1961 was thinking about when she coined the phrase paradox of the plankton. So what she was referring to was phytoplankton that live in seemingly really simple environments. So they live in kind of open water, the open water of freshwater lakes. So they compete for the same nutrients in those lakes. They live in what seem to be completely uniform environments. But yet many species of those phytoplankton coexist, and there's no evidence for competitive exclusion in those communities. So how is that possible? How can so many species exist when they appear to be, on the face of it, occupying basically the same niche? In 1977, David Tillman tried to address that question by growing two competing diatom species, Astrionella and Cyclotella, in tanks with varied ratios of silica and phosphorus. So diatoms are the most common form of phytoplankton in freshwater lakes. They're single cellular photosynthetic organisms coated in a layer of silica. So if you're walking along a lake shore and you find the rocks under your feet to be really slippery, often that's because they're coated in diatoms that have this kind of silica coating around themselves. What Tillman found was that Astrionella was much better than Cyclotella at absorbing phosphorus. So when phosphorus was low in the environment, or phosphorus was limiting, Astrionella would absorb all of that phosphorus too quickly for Cyclotella, and it would exclude Cyclotella completely from the environment. But as the ratio of phosphorus in the environment increased, Astrionella would become slowly limited by silica, and that would mean this would stunt their growth to the point that Cyclotella could be incorporated in the population and could be successful in that environment. And as the rates of phosphorus got higher and higher, eventually in really high ratios of phosphorus to silica, Cyclotella actually became the dominant species. What this experiment shows really nicely is that the outcome of competition between species is dictated by environmental conditions. So what David Tillman's work showed is that the niches of diatoms are defined by their nutrient requirements and their ability to uptake nutrients. So although the environment might seem really uniform, any variation in nutrient availability is going to create different levels of competition and allow different species to thrive in those areas. And Tillman collected water samples from across Lake Michigan. And what he found from those samples is that the silica to phosphorus ratios across the lake range from 1 to 600. So this massive variation in ratios of nutrients mean that in fact, although this environment, the lake, looks really uniform, there are a huge diversity of niches that occur within that lake. And we're at this point only looking at two different nutrients. So when you look at the ratios of all sorts of different nutrients and light and other factors across that lake, the different niches that occur in that lake are actually endless. So David Tillman's work on Lake Michigan highlights that heterogeneity in environments can take many forms. It can be really obvious, so you could have the elevational gradient of a mountainside, or you could have the obvious diversity in plant life of a tropical rainforest. But actually, even in areas that seem pretty uniform, you can get this huge amount of heterogeneity at different levels. So we saw that in the nutrient availability in different parts of Lake Michigan. And it's that variation that supports these many, many phytoplankton species. But here we've got an example of a similar thing on terrestrial ground. So this is an abandoned agricultural field in Michigan, and researchers at Michigan State University measured on this field nitrate levels and moisture levels across the field in a kind of grid of sample sites. And what they found was just this really uneven and seemingly random distribution of nitrate availability and moisture. And if you compare those two maps, you see very little overlap or very little similarity between them. So you can layer those two and get this almost endless combination of nitrate and moisture availabilities. And it's this level of heterogeneity that drives huge diversity in plant species. So in this last section of the lecture, I want to talk about why studying community structure is important. And when it comes to applying our knowledge of ecology, often it comes down to human interest. So ecosystems provide what we call ecological services. So these are the processes and resources provided by ecological systems that are of value to humans. So for example, ecosystems of course are involved in biomass production. So growing crops or growing food, fuel plants, whatever it might be. Ecosystems are important for nutrient cycling, for water filtration, for the sequestration of carbon. These ecosystems provide recreation opportunities for humans. Communities of organisms and insects particularly provide pollination for crops. So any change in community structure that occurs affects the degree to which communities and ecosystems can provide these services. So of all of these ecosystem services, arguably the most important is biomass production. So this is central to the provision of food for human populations. And a big question that's been addressed over many, many years by ecologists is, how does plant diversity affect community productivity? So what we mean by this is, if we increase the number of plant species in a community, how does that affect the overall growth rate or the overall productivity of that community? And that question's been addressed by a couple of huge landscape scale experiments that have been conducted side by side in Minnesota. And this picture shows one of those experiments. So all of these plots that you can see are different communities made up of different numbers of species. And there's an overall pool of species of about 16, and then those same species are grown in different combinations in different numbers of species in all of these different plots. And then over a period of years, the productivity of these different plots has been measured. And the two graphs on this slide show the results of those two different experiments in Minnesota that have been run side by side. So on the x-axis you have planted richness, so that's the species richness of the communities, and then on the y-axis you have relative yield, so the productivity or the overall growth rate of those communities. So what they show is that as the number of species in a community, so the diversity increases, so does the relative yield or productivity. And the reason there are many lines on these graphs is that each line represents 1 year in the study. So they're on a color scale ranging from red to blue. So what it shows is that very quickly species richness led to greater productivity in these sites, but the size of that effect got larger and larger over every year. So the early years are represented by red, and the most recent years are represented by blue. So the longer you leave those communities, the more that increase in diversity affects productivity or increases productivity. So ecologists now are more or less in agreement that increased plant diversity leads to increased productivity. But in terms of what causes this relationship between diversity and function, there's still a lot of debate and a lot of work being done. So there are three main theories. The first being complementarity. This is the idea that a more diverse plant community should be able to use resources more completely and thus be more productive. So in a community where there are more species, those different species are going to use resources differently. So if you look back to David Tillman's diatoms, he just studied two different species and saw how wildly different they were in the ways that they use silica and phosphorus. So by having more species that use resources in different ways, some of those species are going to utilize resources that others wouldn't be able to, perhaps because they're limited by other nutrients. And so this is the idea of complementarity. The second idea is facilitation. This is where the presence of one species enhances the growth or productivity of another. And there are many potential examples of this, but one that immediately springs to mind is that of epiphytic plants. So epiphytes that grow on the surface of other plants. So these plants, if they grow, for example, in the canopies of trees, are going to have much more access to sunlight than they would if they grew on the ground. And so this just means that the presence of this large tree massively enhances the growth or productivity of this epiphytic plant. And the third idea is sampling effects. So this is the idea that some species are naturally more productive than others, certainly true. So as diversity increases, so do the odds that the community contains at least one of these highly productive species. So the idea here is that increased productivity is not actually the result of diversity itself, but just the increased probability that the community now contains highly productive species. So when we look at those three different ideas, complementarity, facilitation, and sampling effects, one thing to certainly note is that those three ideas are not mutually exclusive. So the answer here, as is the case with many scientific questions, is probably a combination of all of these things.